guys, today we're going to be talking about biological molecules. We'll start with a little bit of an introduction and then we'll look at the 4 types of biological molecules we find in living systems: carbohydrates, proteins, lipids, and nucleic acids. Carbon is the predominant element found in living systems. So let's have a look at that carbon atom. Carbon has an atomic number of 6 and an atomic mass number of 12. So this means that inside that nucleus we have 6 protons as well as 6 neutrons. Circulating around the nucleus we find 6 electrons, 2 in that first shell and 4 in our valence electron shell. This means that carbon has 4 vacancies and it can form up to 4 covalent bonds, which in turn means that carbon can bond to a maximum of 4 other atoms. In our organic molecules or biological molecules, we always find at least one carbon atom bonded to a hydrogen atom. Large biological molecules are composed of smaller biological molecules, and those smaller molecules are known as monomers. So if we're building up these larger biological molecules, we use a process known as dehydration synthesis. Dehydration, if you look at that word, hydration talking about water, so we're going to lose water in that process. So for example, over here we have one monomer plus another monomer. We now form a dimer. Remember, di means 2, and we've lost water in the process. If we add another monomer now to that dimer, this is when we form a polymer. Poly means many, and so as soon as we get bigger than 2, we call it a polymer. On the other hand, if we're going to break down larger molecules, we're going to use a process known as hydrolysis. So hydro, once again incorporating water, and lysis meaning to split. So in order to split those larger molecules, we have to have that input of water. So the example here, if we took a dimer, so di meaning it's composed of 2 monomers, we need water in order to split those monomers apart, and we can form a monomer plus another monomer. So to try to remember the difference between these two, let's pretend you're going to build a house. All right, to build this house, you're going to sweat a lot, so you're going to dehydrate. So to build larger macromolecules, we use dehydration synthesis. Whereas if a flood came, a flood can break things down. So we have this influx of water that ends up breaking things down. So that is hydrolysis. The cool thing about this is that we can have a small number of monomers and put them together in different ways to build many different types of polymers. Think about it in terms of playing with Lego. If we have a small number of Lego blocks, we can arrange those blocks in different ways and form different things, such as a house or a tree. So for example, over here, we have these 6 different monomers, and they differ from one another. And we can arrange them in different orientations. We can also arrange them in different orders, and by doing that we can form a number of different polymers. In this case, just with those 6 monomers, we can form 15,625 different polymers. Now if you add to that the fact that we can also change the length of these polymers, the amount of different polymers we can produce is almost infinite. The 4 most abundant types of biological molecules we find in living systems are carbohydrates, lipids, proteins, and nucleic acids. So let's have a look at each of these individually. Firstly, carbohydrates. If you think about the name, carbo referring to carbon, hydrate referring to water. So a carbohydrate is always going to have carbon, hydrogen, and oxygen in the ratio of 1:2:1. We get 2 main types of carbohydrates. We get simple carbohydrates and then our complex carbohydrates. Simple carbohydrates are going to be our sugars, so things like glucose or sucrose, and these are going to be used for short-term energy. Our complex carbohydrates are going to include things like starch and cellulose, and these are going to be used for structure as well as energy storage within the cell. So let's talk about those simple carbohydrates, our sugars. We have 2 types of simple carbohydrates. We have monosaccharides and disaccharides. Our monosaccharides are going to be composed of 3 to 7 carbons. Something like glucose or fructose would be considered a monosaccharide, and it can be present as a straight chain or as a ring structure. Our disaccharides are going to form when 2 monosaccharides join together. So let's have a look at this example over here. We have glucose, which is a monosaccharide, and fructose, which is a monosaccharide. If we had to look at that ratio of carbon, hydrogen, and oxygen in each of these molecules and we simplified it down, it would be 1:2:1. Now, in order to join these two together, we have dehydration synthesis occurring, and we lose hydroxide from glucose, hydrogen from fructose, which is essentially the loss of water. We join them together here at that oxygen atom, and now we form this disaccharide, sucrose. Sucrose is our common table sugar that we use. Sucrose can also be broken back down into glucose and fructose through hydrolysis, and you can see that in order for it to occur, we have to input water because we need that hydroxide ion to come back onto glucose. We need that hydrogen ion to come back onto our fructose. Our complex carbohydrates are our polysaccharides. So they contain many monosaccharides connected to one another. Cellulose is an example of a plant polysaccharide. We find it in the cell wall of the plant. It's going to provide strength and rigidity. And basically what we have in cellulose is a whole bunch of glucose molecules connected to one another. Another plant polysaccharide we find is starch. So starch is a storage molecule and plant used to store energy. If you look at that starch molecule, once again we have glucose molecules linked to one another, but you can see that this molecule is a highly branched molecule compared to the cellulose molecule, which is a straight chain. Last example over here is glycogen. This is a storage molecule that we actually find in humans. And you can see once again we have glucose molecules linked together, and it's a highly branched molecule as well. Another animal example of a polysaccharide would be chitin. We find this in the exoskeleton of our insects. We actually also find chitin in fungi cell walls. Okay, let's move on to our proteins. The monomer of proteins is the amino acid. So let's have a look at the structure of the amino acid. Amino acids have a central carbon atom. Remember that carbon can form 4 bonds. So this carbon atom is going to always be bonded to a hydrogen atom. The other thing we'll find in our amino acid is that it's connected to a carbonyl group, which contains carbon double bonded to oxygen, and then that carbon is also bonded to a hydroxide group. This central carbon atom is also going to be joined to an amino group. The amino group contains nitrogen bonded to 2 hydrogen atoms. This last group over here we call the R group, and that R group is what changes from one amino acid to the next. So we have 20 different possible amino acids. All 20 will have exactly the same components, except for this R group will be different. This table lists all 20 amino acids. If you look carefully, you'll see in each of them the central carbon atom bonded to a hydrogen atom, bonded to your carbonyl group, bonded to your amino group. What's changing? The R group in each of them. This R group determines the chemical properties of that amino acid. So we'll see that some amino acids are going to be non-polar, some will be polar, some will be positively charged, and others will be negatively charged. The physical and chemical properties of our R groups are essential because they determine how our protein will fold, as well as what other molecules our protein will be able to interact with. Amino acids are going to link to one another using a peptide bond, which is a type of covalent bond, and this is going to form a polypeptide chain. Our polypeptide chains will differ from one another in terms of the sequence of amino acids as well as the length. Protein structure is extremely important for its function. When building a protein, the first thing that will happen is amino acids will be connected to one another to form a polypeptide chain. This is known as the primary structure of the protein. The next thing that will happen is amino acids that are close to one another will start to interact with one another, folding that protein up, and this will form our secondary structure of the protein. Amino acids located further away from one another will then start to interact with each other. These interactions are made possible by the chemical and physical properties of the side chains present on the amino acids. This further folding results in a tertiary structure of the protein. This tertiary structure is vital for protein function, and therefore any factor that can affect the structure of the protein will also affect the function of the protein. These types of factors include things like temperature, pH, and salinity. For example, at very high temperatures, the bonds between our amino acids will start to break, and the protein structure can start to unravel. We can either get partial unraveling back to the secondary structure, or the protein could completely unwind back to its primary structure. This process of unraveling the protein is known as denaturation. When denaturation occurs, the protein can no longer function. This is the basis of cooking. So think about an egg. If you take an egg, the white portion of the egg, that is protein. If you go and cook that protein, what are you doing? You're heating it up. High heat is denaturing that protein and breaking it down. Now, when you break it down back to its primary structure, those proteins are no longer soluble in water, and so they start to come out of solution and clump together. And that's why the egg white goes from being that see-through complexion to that hard white complexion. There's actually been some scientists that have been able to uncook an egg. You wouldn't be able to eat it afterwards, but it's still been done. What they did is they added urea to the cooked egg and centrifuged it at high speeds. And what this did is it snapped the proteins back to their tertiary structure. So basically uncooking the egg, going from that cloudy white appearance back to the clear appearance of the egg whites. Proteins are characterized by their function. We have some proteins that are used for storage, as in the case of the egg. We have some proteins that are structural, so for example, our keratin in our hair and nails. We have some proteins that are used for transport, for example, hemoglobin. Hemoglobin is a protein found in our red blood cells that's able to bind oxygen and transport that oxygen around our bodies. We also have proteins that are essential catalysts. A catalyst is a molecule that is able to speed up a chemical reaction but is not consumed in the reaction itself. These types of proteins are specifically called enzymes. Let's move on to lipids. Lipids are hydrophobic molecules. This means that they cannot associate with or dissolve in water. Lipids are also energy rich. Most lipids are composed of one or more fatty acids. Fatty acids are long hydrocarbon chains, and we have two types, saturated and unsaturated. In our saturated fatty acid, every carbon within that chain is saturated with the maximum amount of hydrogen. In our unsaturated fatty acids, we find double bonds. For example, over here we have a double bond between these two carbons. So they're not saturated with hydrogen. If we broke that double bond, we could in fact add another hydrogen to each of those carbon atoms. Saturated fatty acids tend to be straight chains, and they can therefore pack very closely to one another. Because of this, they are generally solids at room temperature. Things like butter would be an example of a saturated fatty acid. However, our non-saturated fatty acids have a kink in the chain because of this double bond. That kink prevents them from packing very closely to one another. And because of that, they are generally liquids at room temperature. So an example there would be olive oil. Fatty acids are normally joined to glycerol. So if we have a look at glycerol, you'll see we have 3 carbon atoms bonded to hydrogens and oxygen. The glycerol molecule could have 1, 2, or 3 fatty acid chains attached to it. The most common type we find in our diet is where we have 3 fatty acid chains attached, and this is known as a triglyceride. I've already mentioned that animals use the carbohydrate glycogen to store energy, but in fact, the most common storage molecule in animals is a triglyceride. The reason for this is that triglycerides take up less space in the cell. Carbohydrates are polar, so they interact with water, and if they're in the cell, they're going to be associated with a whole bunch of water molecules taking up more space. Fats, on the other hand, are hydrophobic. They cannot interact with water, and so they take up less space in the cell. A very important lipid within living organisms is the phospholipid. This consists of a phosphate head group along with 2 fatty acid tails. The phosphate head is hydrophilic, which means that it can interact with water. The fatty acid tails are hydrophobic and cannot interact with water. When placed in water, these phospholipids are going to form a lipid bilayer, with the heads on the outside where they can interact with water, and the fatty acid tails sequestered on the inside far away from any water. These phospholipids are essential components of membranes that we find inside the cell. Another lipid we find in living systems is sterol. Sterol is going to be composed of 4 hydrocarbon rings connected to one another. Sterols will differ from one another in terms of the functional groups attached to these 4 hydrocarbon rings. Examples of sterols include testosterone, estrogen, vitamin D, and cholesterol. Our next type of biological molecule we're going to look at is the nucleic acid. The monomer for nucleic acids is the nucleotide. Looking at the structure of that nucleotide, we see that it has a pentose sugar. Pentose meaning containing 5 carbon atoms. There are 2 different types of pentose sugars we find. We find ribose or deoxyribose. Attached to that sugar is a phosphate group as well as a nitrogenous base, meaning it contains nitrogen. And we have 5 different possible nitrogenous bases: adenine, thymine, cytosine, guanine, and uracil. Our monomers or our nucleotides are going to connect to one another to form a polynucleotide chain. And this connection is going to be between the phosphate of one nucleotide and the sugar of the next nucleotide in that polynucleotide chain. Once again, this is going to be a covalent bond. Nucleic acids are the molecules that are going to store and transmit genetic information within the cell. And we have 2 types. We have DNA and we have RNA. DNA is the storage molecule. It's going to contain the sugar deoxyribose as well as the nitrogenous bases adenine, thymine, cytosine, and guanine. Our DNA is going to be double-stranded. And so what that means is we have 2 polynucleotide chains connected to one another, and that connection occurs at the nitrogenous bases. We have what we call base pairing rules, so adenine and thymine are always bound together, whereas guanine and cytosine are always bound together. Our RNA, on the other hand, enables the cell to use that information that's locked away in the DNA. The RNA is going to have the sugar ribose, it's going to be single-stranded, and it's not going to contain thymine. So instead of thymine, it has uracil along with adenine, guanine, and cytosine. A very important nucleotide within cells is ATP, adenosine triphosphate. This is an energy carrier and it's able to transfer energy from one reaction to the next. If we have a look at the structure of ATP, we have the sugar ribose, and that ribose is connected to the nitrogenous base adenine. Now, instead of having a single phosphate group, we actually have 3 phosphate groups attached to that sugar. And this is where our energy is stored. So energy is stored between these phosphate groups, and it's going to be released when those phosphate groups are cleaved from the molecule. So ATP is high energy. If we release this last phosphate, we form ADP, adenosine diphosphate, which has low energy. This molecule is unstable. And the reason for that is because these bonds are such high energy, they're easily broken. Also, we have these negative charges on the oxygen molecules which are repelling one another, once again making those bonds easily broken. So it's important actually that this molecule is unstable because the energy is released easily and made available to the cell. On the other hand, the downside is that we constantly have to make more ATP to fuel the different metabolic reactions within the cell. We've come to the end of our session, and I'd like to encourage you to go back, use the video as well as the slides to answer these review questions and make sure that you understand.